I welcome you all to a new session of A Path to Success. I am Dr. Induyam, Assistant Professor, Government Dental College, Kottayam. Today, I'll be dealing with the topic genetics. We look so different from each other in terms of our skin color, hair type, height, weight, etc. And we look similar to our parents. What is the reason behind this? The reason behind the differences are variations. The variations are the degree to which the offspring differ from their parents. And similarities can be explained with the help of inheritance. Inheritance is a process by which the characters are passed from parents to their offspring. The variations and inheritance are controlled by genes, and study about a single gene or a group of genes is called as genetics. Today, we will discuss more about inheritance. Before moving on to the details of inheritance, let us discuss about the basic structures like chromosomes, DNA, and gene. Chromosome is a coiled structure or a colored body seen within cellular nucleus. It is actually made up of DNA supercoils around proteins called as histones. So if we unwind a chromosome, we can see that the stretch of chromosomes wind around histone proteins to form a structure called as nucleosomes. The DNA is a basic genetic material for most of the organisms, and a stretch of DNA can make a gene. Gene is a basic unit of heredity, and it contains information about a particular trait or a character. The structure of a chromosome. A chromosome contains two identical pairs of threads, which is called as chromatids or sister chromatids. And these chromatids are joined at a primary construction called as centromere. And each chromatid can be divided into a short arm called as P arm and a long arm called as Q arm. And tips of the chromosome will be capped by telomeres. And the number of chromosomes differ in each species. In human beings, there are 23 pairs of chromosomes. In that, 22 pairs are autosomes and one pair is a sex chromosome. So for a female, the genetic constitution is XX, and for a male, it is XY. If we want to locate a particular gene in a chromosome, we should know about the arm, region, band, and subband. So imagine this is a chromatid, and it has a short arm and it has a long arm. And in this short arm, there are two regions, 1 and 2. And again, the region can be divided into bands. So here, region 2 of short arm of this chromatid has band 1 and band 2. But the region 1 of this long arm has band 1, band 2, and band 3. And if we are using high resolution imaging, we'll get the sub-bands also. So if we want to locate a gene or write the position of a gene, you should know the chromosome number, the arm, the region, and the band. DNA. DNAs are polymers of nucleotides. The nucleotide contains phosphate moiety, sugar molecule, and a nitrogen base. So here, the two chains of nucleotide polymers are arranged in a double helical pattern, and these two chains are anti-parallel to each other. The backbone of the DNA is made up of the sugar and phosphate moiety. Okay, and the center portion of the helix contains nitrogen bases. So this nitrogen bases can be adenine, thymine, guanine, and cytosine. Adenine will always bind with thymine, and cytosine will always bind with guanine of the adjacent chain. So adenine is held together with the thymine with the help of two hydrogen bonds, and guanine and cytosine are binded by three hydrogen bonds. Gene is the basic unit of heredity, and the structure of a gene we can divide into promoter region and the coding region. The promoter region controls the transcription process, and the coding region help in formation of mRNA. If you examine the region, you can make out that the exon region are not in continuous stretch. The exons are actually the coding region which help in formation of mRNA. In between the exon, there are intronic regions. The intronic regions are non-coding region, which will be spliced out during the formation of mRNA. And then you should know about a few more terminologies like alleles, phenotype, and genotype. So what is alleles? Allele is an alternate form of a gene, and that is located in a specific chromosome in a specific position. Alleles always occur in pairs. In a diploid organism like us, each character is controlled by a pair of alleles. For example, here the character is height, and it is controlled by a pair of alleles like capital T, capital T, capital T, small t, and small t, small t. Phenotype. Phenotype is the observable physical characteristic of an organism. In this group of plants, they differ by the character of height. So it is a phenotypic character. Then genotype is the allelic constitution that underlies behind that particular phenotype. So let us examine the allelic constitution of this plant. The dwarf plant has alleles like small t, the tall plant capital T, and capital T, small t. So this is the genotype of this plant. Another example, blood group A, B, AB, and O. They are phenotype. And behind this phenotypic expression, there are 6 genotypes. A significant level of understanding about inheritance was obtained by the experiments conducted by Father Gregor Mendel, and he is the father of genetics. So Father Gregor Mendel has conducted hybridization experiments on garden pea plant. And on the basis of these experiments, he proposed laws of inheritance. And these laws are law of dominance, law of segregation, and law of independent assortment. Law of dominance. In this group of plant, one plant is dwarf and other plants are tall. Its alleles for the dwarf plant is small t, and alleles for this tall plant is capital T, And in this condition, both alleles are similar. So we can call it as a homozygous condition. Here, for this tall plant, alleles are capital T and small t, both alleles are dissimilar to each other. So this condition is called as a heterozygous condition. So in this heterozygous condition, character of the small t is masked and character of this capital T is expressed. Okay, the character of the small t is the dwarfism, but here the plant is tall. That means the character of capital T is expressed. So the capital T dominate over the small t. That is, the small t is a recessive allele and capital T is a dominant allele. So law of dominance states that the characters are controlled by discrete units called as factors or genes. And these factors pass from one generation to other generation and they occur in pairs. Here, the alleles occur in pairs. And in a dissimilar pair of factors, one factor dominates over the other. In this dissimilar condition, okay, there's a capital T and small t, one allele dominate over the other allele. Father Gregor Mendel has conducted a monohybrid cross experiment which support law of dominance. Monohybrid means two group of organisms which differ with respect to one character. Here, this plant is tall, this plant is dwarf. It has alleles capital T, dwarf plant, small t, small t. As we know, capital T is a dominant allele and it will be expressed even in heterozygous conditions. And small t is a recessive allele and its character is masked in heterozygous condition. It will only expressed in homozygous condition. So during hybridization, the dwarf plant will produce gametes with alleles small t. Here from here, capital T, capital T. So these will combine and produce F1 generation. So all plants in the F1 generation are in a heterozygous state, capital T, small t. Okay, so the capital T is a dominant one. So all plants in the F1 generation will be tall. So during self-pollination of this F1 generation, this tall plant with the alleles capital T, small t will produce gametes with containing capital T allele and small t alleles. And during hybridization, this capital T will combine with capital T to produce a homozygous state. Small t will combine with the small t will produce again a homozygous state where the plant will be dwarf and rest are in a heterozygous condition. So in heterozygous condition, the character of the dominant allele is expressed. So there are certain phenomena in the nature which cannot be explained with the help of law of dominance. Such an example is a codominance. So what is codominance? Codominance means in a heterozygous relationship, both alleles are completely expressed. So this is a 4 o'clock plant. Okay, this plant has alleles for yellow color petal and pink color petal. So both alleles are expressed completely. So this flower has both yellow color petal and pink color petals. Another example is AB blood group. The AB blood group has allele A and allele B. Both alleles are expressed completely. The next law is the law of segregation. So here also a monohybrid cross of the plants are used. So this is a tall plant with capital T, capital T. This is a small dwarf plant with a small t, small t. So during gamete formation, the gamete will receive one capital T allele from the parent plant. Here, one small gamete will receive one small t allele from the parent plant. So during hybridization, this capital T will combine with the small t's and produce a heterozygous group of offsprings. So all offsprings has allele combination capital T, small t, and all these plants will be tall. So in the F1 generation, the plant has alleles constitution capital T, small t. So what will happen during gamete formation? During gamete formation, these alleles will segregate each other, the capital T and small t will segregate each other, so that the one gamete will receive either capital T or the small t allele. Okay, so during self-pollination, it will produce a combination of plants in which one plant is dwarf and other plants are tall in the ratio 3:1. So from this, we can conclude that the parents contain two alleles, and alleles of a pair segregate from each other such that a gamete receive only one of the two factors. Here, the parent contains a pair of alleles and it will segregate from each other, and the gamete receive only one of the alleles. Next is law of independent assortment. Here, Mendel has used a dihybrid cross. That means the plants or the organism differ with respect to two characters. Here the plant seed is round and yellow. Here it is wrinkled and green. So there is two characters. They are differing. One is a shape and other is a color. So this plant has allele constitution capital R and capital Y. This plant has allele constitution small r and small y. So during gamete formation, one gamete will receive one capital R and one capital Y from this parent plant, and here the gamete will receive one small r and one small y from this parent. So during hybridization, these gametes will combine with these gametes to produce a heterozygous group of offspring. So all the offsprings will have a genetic constitution of capital R, capital Y, small y. Okay, so this yellow character and round character are dominant one. So all the plants in the F1 generation produce round and yellow seeds. Okay, so these round and yellow seeds, they have allele constitution capital R, capital Y, small y. So how will be the gamete formation? 50% of the gamete will receive this capital R. Other 50% of the gamete will receive this small r allele. And again, 50% of allele will receive capital Y and 50% of allele will receive small y. Okay. And this shows that segregation of 50% of capital R and small r are independent of segregation of capital Y and small y. That means segregation of one character is independent of segregation of other character. In the F2 generation, we can make out 4 different phenotypes and 8 different genotypes. And from this experiment, we can conclude that when 2 pairs of traits are combined in a hybrid, segregation of one pair of character is independent of segregation of other pair of character. This is the hybrid condition, and segregation of alleles controlling the shape is independent of segregation of alleles controlling the color. Types of inheritance. 4 types of inheritance are mainly seen: autosomal dominant, autosomal recessive, X-linked dominant, and X-linked recessive. The autosomal disease means the affected gene is located in any of these autosomes. X-linked disorder means the affected gene is located on the X chromosome. In this family photo, we can make out that all the individuals are short. This is an example of achondroplastic condition. This is an autosomal dominant disorder. There are many other disorders in this category. For example, von Willebrand disease, osteogenesis imperfecta, neurofibromatosis, Noonan syndrome, etc. So we'll see how is the inheritance pattern of an autosomal dominant disorder. So in this example, there is an unaffected parent with a normal allele, small a, and an affected parent with an allele, capital A and small a. The capital A is the diseased allele, which is a dominant one. So the character will be expressed. So the affected parent will produce gametes with the genetic constitution, like one with capital A and the other with small a. So during hybridization, the diseased allele combine with the normal gametes and produce affected individuals. And at the same time, 50% of the offsprings will be normal. So for the affected offsprings, the genetic constitution will be capital A and capital small a. Okay. So 50% of the children are affected here. And here we can make out that one son is affected and one daughter is affected. We can represent this inheritance with the help of a pedigree chart also. So what is a pedigree? It is a family tree or chart which is made up of symbols and lines. It represents the genetic family history and it will help in determination of mode of inheritance. Here we'll discuss a few of these symbols. The square means male, circle means female, a square and circle with interconnected horizontal lines, mating, a square and circle with two horizontal lines means consanguineous marriages. And a square circle interconnected horizontal line with vertical compartments means parents and the children. If the male and female symbols are shaded, it means they are affected. If it is half shaded, it means they are heterozygous for autosomal recessive disorder. If a dot is present in the center, it means they are carriers for sex-linked recessive disorder. If there is an arrow with a square or with a circle, it means they are the proband or they are the first deceased individuals in the family noticed by the healthcare professionals. Here comes the pedigree chart of an autosomal dominant disorder. Here father is normal with a normal allele constitution A and mother is affected with a diseased dominant allele capital A and a normal allele. So during hybridization, this diseased allele will combine with gametes with normal allele. So there will be a capital A small a combination in 50% of offsprings. So these offsprings will be affected and these offsprings will be normal. Okay, so from this chart, we can conclude that an autosomal dominant disorder does not skip generations. That means you can identify deceased individuals in all generations. And there is an equal chance for male and female offsprings to get affected. Here in second generation, one son and one daughter is affected. And this disorder can be transmitted by either father or mother. Because this affected gene is located on an autosome, not on a sex chromosome. So there is an equal chance for male and female, and it can be transmitted from father or mother. Autosomal recessive disorder. Familiar examples are sickle cell anemia and cystic fibrosis. So in an autosomal recessive disorder, for example, so father with a genetic constitution capital A small a and mother with a genetic constitution capital A small a. The small a is the disease allele. But the disorder is autosomal recessive. So in a heterozygous condition, this recessive allele will not be expressed. So both these parents are carriers. So during hybridization, one parent will produce a normal copy of allele and a gamete with a diseased copy. Same here. So during hybridization, this capital A will combine with capital A to produce a normal offspring. Small diseased allele will combine with the diseased allele to produce an affected individual. So this is a homozygous state. So here this individual will be affected, and other offsprings are in a heterozygous state. Okay, so they will be carriers. So in this example, in second generation, one son is affected with a homozygous state, small a, both alleles are diseased. One daughter is affected, small a, and others are carriers, and one offspring is normal. Okay, in the third generation also, one son is affected with the homozygous disease state, and one daughter is carrier. So from this chart, we can make out that the autosomal recessive disorders skip generation. So some generations may not have diseased individuals. In the first generation, both parents are carriers. Then there will be equal chance for males and females to get affected, and the disease can be transmitted from father or mother, because these are autosomal disorders. And most affected individuals will have unaffected parents. Here, the son is affected and he has unaffected parents. They are phenotypically normal. Another condition in autosomal recessive disorder, if both parents are affected, that means both parents are in homozygous state. Okay, so their constitution will be small a, small a. So all the gametes produced from them will be affected. They'll contain diseased allele. So all their parents will get affected by this particular disorder. X-linked dominant disorders. X-linked means a particular gene will be located on the X chromosome. A common example, a familiar example, is vitamin D resistant rickets, in which the long bone will be soft and bent. And other disorders are Rett syndrome, Fragile X syndrome, Alport syndrome, etc. They are comparatively rare disorders. We'll see the inheritance pattern of an X-linked dominant disorder. Here, the father is affected with a defective X copy and normal Y copy. The mother will have a normal X chromosome, 2 copies of normal X chromosome. So what will happen? The father will have a defective X copy, and mother will produce gametes with the normal X chromosome. So this defective copy will combine with the normal X chromosomes to produce defective individuals or affected offsprings. So these offsprings are females. They have an XX combination. Okay, because the disease is dominant one, they'll get affected. And the sons, they'll receive a normal Y chromosome from the father and a normal X chromosome from the mother. So the sons will be normal. So in this example, we can make out that this affected father will have all his daughters as affected and all his son will be unaffected. And another example, the father is unaffected and mother is affected. The mother will have one defective X copy. So during hybridization, the father will produce normal gametes and mother will produce one defective copy. So this gamete with this affected allele will combine with the normal gametes to produce a diseased female offspring and a diseased male offspring. And here also 50% of individuals are affected. So in an X-linked dominant disorder, 50% of the offsprings will be affected. And from these findings, we can conclude that X-linked dominant disorder does not skip generations. And the affected male always comes from affected mother. So here there is an affected male. So this affected male receive normal Y chromosome from this father and an X chromosome from this mother. So he can receive this defective X chromosome from the mother. So the son will be affected. The affected female come from affected father or affected mother. The affected female, this is an affected female. So she can receive the affected X chromosome from the mother, or she can receive the defective X chromosome from the father. Half of children of affected mother will be affected. So this mother is affected and half of her children are affected because she'll produce 2 copies of X chromosome. One is defective. Okay, so 50% of the kids will receive this defective copy and another 50% of the kids will receive this normal copy. And all daughters of affected father will be affected. Here the father is affected and the daughter will receive 1 X chromosome from the father and 1 X chromosome from the mother. So all daughters will inherit this defective X chromosome from the father and they will be affected. And at the same time, the sons who receive the normal Y chromosome from the father, so they will be unaffected. So this is a pedigree chart of X-linked dominant disorder. We can say that there are deceased individuals in all generations. Okay, and here there is an affected male and all daughters of this affected male is affected and all sons are unaffected. X-linked recessive disorder. The common examples are royal disease or hemophilia and the color blindness. And we'll see the pattern of inheritance. Here, the father is affected and mother is normal. Father has a defective X copy. So during hybridization, the gamete with affected X allele will combine with normal gametes from the mother and produce carrier individuals. Why carriers? Because it is a recessive condition and the trait will be expressed only in the homozygous state. So these 2 offsprings are females. They have 2 X chromosomes. So this unaffected X chromosome will dominate over the affected X chromosome. So they'll remain as carriers. The sons who receive normal Y chromosome from the father remain unaffected. So for a deceased or affected father, his daughters will be carriers and sons will be unaffected. Next example, father is unaffected and mother is carrier. So mother has 1 defective copy. So during hybridization, father will produce normal gametes and mother will produce a gamete with normal X chromosome and affected X chromosome. So this affected X chromosome combined with normal X and normal Y chromosome and these individuals shows variations. So here it is a female in offspring. She has 2 X chromosomes. So she'll be a carrier. Here it's a male. The male has only 1 X chromosome and this X chromosome is affected. So this male will be affected with the disease. So from this example, we can make out that the most commonly affected offsprings are males. Coming to the features of X-linked recessive disorder, most of the affected people are males because they have only 1 X copy. And affected males always come from affected or carrier mothers because they receive X chromosome from the mother and Y chromosome from the father. And sons of a carrier females. 50% of sons of carrier female will be affected. The carrier female will produce gametes with a normal X copy and gametes with a diseased or abnormal X allele. So 50% of her sons will be affected and 50% of her sons will be unaffected. Another 2 features of X-linked recessive disorder, sons of affected females will be affected. Affected female means they are in a homozygous condition. They'll produce gametes with the defective alleles. So the sons will receive X chromosome from the mother and the sons will be affected. Then an affected female comes from affected father and affected or carrier mother. If a female has to be affected, she has 2 copies of X chromosome. The both copies should be affected and should be in a homozygous state. Otherwise, she'll become a carrier. So this affected individual receive 1 X copy from father, 1 deceased X copy from the father. The father is affected. The second X chromosome she'll receive from mother. So mother should be either carrier or affected. This is a pedigree chart of a hemophilia disorder. Here in the first generation, 1 female is a carrier. So she passed her defective X copy to her sons and daughters. So daughters remain carriers and the son is affected. Again, in the next generation, 2 male offsprings are affected. And if we examine this pedigree chart, it's clear that the male offsprings are affected by the disease and females remain carriers. There are other modes of inheritance also, like Y-linked inheritance and mitochondrial inheritance. They're comparatively rare. And I would like to introduce 2 more terminologies. One is penetrance and other is expressivity. Consider this group of people has same genetic constitution of capital B, small b. The capital B represent a diseased allele and it is dominant. And in this group of people, only 5 of them expressed the phenotype or 5 of them express the disease and 5 of them appear normal, even if all of them have same allelic constitution. That means 5 out of 10 individuals expressed the phenotype or the disease. So the penetrance is 50%. So what is a penetrance? Penetrance of an individual with it's a percentage of individuals with a given allele who can exhibit the phenotype. Here only 50% of the individuals express the phenotype. So penetrance is 50%. So some alleles will have complete penetrance. That means all individuals with that particular allele will express the phenotype. Some alleles will have variable penetrance or incomplete penetrance. That means only few individuals will express the phenotype. Next is expressivity. Consider this group of people with the same genetic constitution, capital B, small b, and this is the deceased allele. Okay, so out of 10 individuals, 3 individuals express severe symptoms. And 4 individuals express moderate symptoms, and 3 individuals express mild level of symptoms. So even if the genetic constitution is same, the intensity of expression differs. So expressivity means it is the degree to which the given allele is expressed at the phenotypic level. So it is the intensity of the phenotype or the disease. This is a common example for expressivity. All these dogs has same allelic constitution. Okay, but their spotting pattern has differ in its intensity. So they show varying expressivity. So in a group of people, we can see varying penetrance and varying expressivity together. And by this, I would like to conclude today's discussion and hope we'll discuss about variations and abnormalities in another session. Thank you.